In this penultimate segment of our video seminar series, I'll try to give voice as fairly as I can to some of the objections raised to our inferences on mitochondrial elevated temperature. To kick off, I need to state that I am not a theoretical physicist, and thus do not feel fully qualified to comment on the detailed arguments of those who are. Merely, I can report them. To position this video in the series, it's worth recapping where we are. In past segments, we've reviewed the evidence for elevated mitochondrial temperature and looked at the implications for cell biology, evolution, and most recently disease. We looked at the possibility that mitochondrial DNA evolution has contributed both to environmental adaptation but also maladaptation in humans, at how mitochondrial disease has been found to be associated with both hyperthermia and hypothermia in specific cases, how its symptoms are often initiated by episodes of infection and high fever, and how the thermal output of malfunctioning mitochondria might create a vicious cycle of overheating and organ damage in the heart. So in this part of the series, I'll look at some of the starting assumptions, some of the objections and caveats to our findings. Can mitochondria really be sustained at a much higher temperature than their surroundings, the cytosol? What does physics teach us about sharp temperature differences between compartments in the cell? And what can studies of intracellular architecture contribute to the debate? We'll next look at evidence that the OXPHOS complexes and supercomplexes may themselves be thermally unstable. We will then consider in this slide the physiological roles of the many subunits present in the mitochondrial OXPHOS complexes but absent from the bacterial counterparts, and the peculiar case of tunicates. The first thing that we need to consider is what the various mitochondrial temperature probes used by ourselves and others actually measure. While calibrations show clearly that they are responsive to temperature, could the readouts from MTY and GTemp also be influenced by other parameters? As we discussed back in part 2, our attempts to identify any such confounding factors, at least for MTY, were negative. But that doesn't mean that there are some that we or our peer reviewers never thought of. Some have also argued that Fourier's law precludes such large temperature gradients across the tiny organella boundaries. But I believe this argument is based on a fundamental misunderstanding of cell architecture. Mitochondria are not simply hot balls floating in a cool cytosol, but their own boundaries are complex and multipartite, with the heat-generating respiratory membrane separated from the inner boundary membrane by much matrix, the inner membrane separated from the outer membrane by an aqueous intermembrane space of at least a few nanometers, and with a perimitochondrial cytosol of unknown structure, size, and composition, as well as other adjacent membranes, such as that of the endoplasmic reticulum, the ER. Temperature gradients across each of these boundaries need to be taken into account. In regard to the perimitochondrial cytosol, we ourselves found that mitochondria behave quite differently when resuspended in an aqueous solution, implying that they essentially depend on the proteinaceous and possibly multiphasic environment around them to maintain their own temperature, essentially an extra insulating layer. And the meltome analysis, to which we already referred in part 2, supports the idea that different subcompartments of the mitochondrial architecture likely operate over distinct temperature ranges. In any case, we need to establish if Fourier's law even applies at nanoscale. What effect do the heat-generating elements, the OXPHOS complexes, have on each other? Moreover, what does temperature even mean at the molecular level? As others have argued, Fourier's law is inadequate for describing heat flows from such closely spaced elements, which must impinge on each other in the same way as those in a microchip. Another consideration we have largely set aside, and which we also touched on fleetingly in part 2, is the heterogeneity of mitochondrial temperature in and between cells. Our fluorescence measurements using a cell population, are really only an average, and include cells with bright staining, cool mitochondria, as well as mitochondria that actually seem to warm up when cells are treated with inhibitors such as oligomycin. Some mitochondria must therefore be well above 52 degrees Celsius if our measurements are an average. As well as this, MTY at least doesn't seem to give reliable measurements in some specific cell lines, notably many human fibroblast lines, for reasons that remain obscure. We also need to be aware that if membrane potential collapses completely, MTY will leach out of mitochondria and then out of cells, so can give an erroneous reading. In studies such as this, it's always advisable to use multiple methods, in our case, both a small molecule dye and a proteinaceous reporter, such as GTEMP. I'd now like to turn to an important question raised in a paper from the Enriquez group in Madrid, published in 2023, that documented the disaggregation of respiratory supercomplexes and even of some subunits of the OXPHOS complexes themselves at temperatures well below 50 degrees, even in isolated mitochondria. The authors interpreted this to mean that reports of elevated mitochondria had to be erroneous. Otherwise, the OXPHOS system would be unable to function. We should caveat this caveat with two important comments before proceeding. First, the actual function of supercomplexes is unclear and disputed. Second, the actual temperature of the mitochondrial complexes is determined partly by externally applied heat, but of course partly by its endogenously generated heat. If the latter produces a steady-state temperature of 52 degrees, an additional external heat source without the insulation normally provided by the perimitochondrial cytosol may simply push this temperature to a much higher level. Of note, we already ourselves found that MTY-stained mitochondria were not able to sustain a steady reading indicative of a high temperature once their external cytosolic insulation was removed. Pierre Rustin's group also found that the optimal operating temperature for the isolated OXPHOS complexes 2+3 and 4 was in the 50 to 55 degree range. The simplest interpretation of the Moreno-Los Huertas paper that they put forward is that the supercomplexes do fall apart at high temperature, implying that the condition where only the individual complexes are detected is a preparative artifact due to a non-physiologically high temperature. But since there is no clearly agreed function for the supercomplexes, it could equally well be the other way around, that the OXPHOS complexes operating as individual units is the natural state of the respiratory membrane at physiological temperature, and that the supercomplexes are a cold temperature artifact. Thinking along these lines, more plausibly, since the supercomplexes seem to be well ordered and retain activity, I suggest that their formation is most unlikely to be an artifact, but instead could be a physiologically programmed response to cold shock, such that respiration can then proceed with reasonable efficiency if a sudden loss of input to the system or other stress with cooling effect is encountered. This makes sense in that the mobility of the small molecular weight carriers, coenzyme Q and cytochrome C, is likely to be decreased if the membrane as a whole cools down below a certain threshold. Supercomplexes may thus be a vital step to maintain efficient respiration in the cold, while at normal physiological temperatures, it's simply unnecessary. In this light, I'd like to turn back now to the differences in the architecture of the OXPHOS complexes between aerobic bacteria and mitochondria, as depicted in the cartoon shown in the slide, that should obviously not be interpreted literally, at least not in fine detail. It's clear, however, that the three core subunits of complex 4 function perfectly well in the bacterial complex without the intrusion of all the others, whose only known role, where specific subunits are represented by multi-gene families, is to optimize the enzyme for different oxygen concentrations. In complex 3, The essential subunits are just the two cytochromes and the Rieske iron-sulfur protein. Indeed, the two so-called core subunits, core 1 and core 2, are close relatives of the two subunits of the major mitochondrial processing peptidase located in the matrix. In some taxa, the same polypeptides are present in both complexes, the peptidase holoenzyme and complex 3. Complex 1 is missing many of its 42 or so mammalian polypeptides, even in fungi, and in bacteria can comprise as few as 14 subunits. The role of almost all of these extra subunits in the mitochondrial complexes of animals remains unknown. Various explanations have been proposed, though firm evidence for any of them really seems to be lacking, except in a few specific cases where physiological regulation has been demonstrated. Unlike the bacterial complexes, 4 of those found in mitochondria are assembled from subunits encoded in 2 different separated genomes, nuclear and mitochondrial DNA, and it's been suggested that many of the extra subunits somehow facilitate assembly under these conditions. Tellingly, however, complex 2, which is entirely nuclear-coded in mammals, is of similar subunit composition in mitochondria and bacteria. Alternatively, many of the additional subunits may facilitate the interactions that lead to the formation of the supercomplexes and the overall organization of the membrane. Another possibility is that they are connected in some way with heat production, but this itself leaves many possibilities worth exploring. For example, the extra subunits might provide extra stability to the complexes in case of transient overheating. Or perhaps they provide thermal insulation to keep the heat in and enable an optimal operating temperature to be maintained. Another possibility, if my hypothesis is correct that supercomplex formation is a response to overcooling, is that the additional subunits undergo conformational changes in the cold that enables their interactions to form the supercomplexes. The extra subunits might also have functional importance at temperatures far from the optimal range. They might serve as a regulatable heat conducting system from the core enzymes to the membrane, or as sensors of thermal signaling. But as yet there is no evidence to support any of these ideas. Deepening the mystery, a recent study by Marcos Oliveira's group in Sao Paulo has found that 15 of those extra subunits and the genes that encode them are completely absent in tunicates, a sister group of the vertebrates. In general, the missing subunits are located on the periphery of the complexes affected, 1, 3, and 4, consistent with the idea that they might be involved with supercomplex formation. One suggestion has been that their absence enables both the complexes and the alternative respiratory enzymes like AOX to interact more easily with the intermediate electron carriers of the respiratory membrane, ubiquinone and cytochrome C. The attractiveness of this idea is that it corresponds with the presence of a complete thermogenic non-proton motive electron transfer chain provided by the alternative enzymes that is theoretically enabling tunicates like Siona to switch the ATP heat balance completely to heat production. It's now time to give a short preview of Part 9, the last in this series of video seminars, where we will take a detour to Antarctica and revisit the question of how organisms adapt to different temperature environments, focusing on a specific example, the case of the Antarctic sea urchin. We envisage a future research project that would require a lot of investment, but for which the basic techniques are already established, which I illustrate with my own lab's earlier work on Siona. It remains for me to acknowledge the contributions of my colleagues and collaborators, as well as that of Tonio Enriquez, whose work on the supercomplexes I have mentioned here. Although we don't agree on the detailed interpretation, Tonio is someone whose opinions and objectivity I greatly value. I should also acknowledge the sources of the various images I've used in this presentation.